Today I'll be talking on the Albert staining. So this Albert staining is a type of a differential stain used for staining the high molecular weight polymers of polyphosphate known as metachromatic granules or volutin granules, which is found in Mycobacterium diphtheria. It is named as metachromatic because of its property which can change the color when stained with blue stain. When stained with blue stain, they appear red in color. When grown in Loeffler slopes, they produces large number of granules. Now, the principle of this Albert stain is made up of two stains that are toluidine blue O and malachite green, both of which are basic dyes with a high affinity for acidic tissue components like cytoplasm. The pH of Albert stain is adjusted to 2.8 by using acetic acid, which becomes basic for volutin granules as the pH of volutin granules is highly acidic. Therefore, on applying Albert stain to the smear, toluidine blue O stains volutin granules, that is the most acidic part of cells, and malachite green stains the cytoplasm blue-green. On aiding Albert iodine, due to effect of iodine, the metachromatic property is not observed and granules appear blue in color. To use this Albert staining solution, each of the two solution must be prepared effectively with the right percentage, or you can buy it. As it is commercially available. So these two solution is needed in order to demonstrate the granules with the right color after staining. As Albert staining solution 1, they act as the staining solution, while Albert's solution 2, they act as the mordant, that is an ion element that binds and holds a chemical dye to make it stuck on the microorganism. So let us see what are the requirements we will be needing to perform this Albert staining. So we will be needing the glass slide and the inoculating loop, then of course the specimen. So for the specimen, you can take it directly from the sample or from the culture plate with grout or from the broth with grout, and also you'll be needing the microscope, and very importantly the Albert A solution and Albert B solution. So let us see the procedure. So in the procedure, I have divided it's one procedure, but I have divided into like two forms, so in order to make it more clear to you all. So the first part is the staining, which uses the Albert staining solution 1, and the second part they uses the Albert staining solution 2. So let us see the procedure. Firstly, aseptically, you have to take a loopful culture of Mycobacterium diphtheria, then make a good smear at the center of a clean sterile glass slide, then heat fix the smear gently. Now on a staining rag, you place the smear glass slide, then add Albert staining solution 1 into the smear and leave it for 3 to 5 minutes. As you can keep it for 7 minutes also, it it differs according to the lab. Then wash the smear slide. So the next step is like the first part we have used the staining Albert staining solution 1. So now we are going to be using the Albert staining solution 2, which is more than thin. So you add Albert staining solution 2 and leave it for 1 minute, then wash the slide with water, then dry the smear glass slide and add cedar oil on the smear and observe under a microscope by oil immersion. So this is the procedure of the Albert staining, where you are going to be using the Albert staining solution 1 and Albert staining solution 2. So let us see the interpretation order result. So here in the interpretation order result, the metachromatic granule stain bluish black, while the rays of the microbial cell stains green. So the interpretation or the result of this Albert stain is like If Corynebacterium diphtheria is present in the sample, it will appear green color in rod shape, which is arranged at an angle to each other and resembling the Chinese letter L or V, and with a uniform arrangement along with the bluish black metachromatic granules at the pole. So these granules um, granules are like they are stained bluish bluish black. This is because of the Albert staining solution B. And the main uses of the main uses of this Albert stain is to help to distinguish the Corynebacterium diphtheria from most of the short non-pathogenic diphtheroid which lack the granules. So this is all about the Albert staining.